Okay, so good afternoon, everyone. Welcome to this class. Uh, so today is the seventh session, okay, of this course, that is cell biology. And so we had six previous sessions, and if you have any doubt regarding any of this, okay, so we can discuss before continuing our lecture. In the previous session, if you have any doubt, so you can ask if you had any doubt in the previous session, so we can discuss. If you don't have, then we can continue with this class regarding the assignment question and all. If you had any doubt, so you can ask. Okay. Is the screen visible to all of you? Yeah. So, anyone have any doubt in the check? Stimulation and ubiquitination in respect to what lysine, sorry, histone modification.
Yes, sir.
Yeah, stimulation and ubiquitination. Yes. So that is ubiquitination in regards to, if I share those slides. So this was the modification which was present in the stone tail, and over there ubiquitination is there, ubiquitin modification. Ubiquitin basically helps uh, uh, to degrade the protein molecule. So ubiquitin mediated mechanism we discussed in the earlier class, that is by proteasome system. Okay, so the ubiquitin molecule, which is composed of three subunit, that is E1, E2, and E3. Okay, so let's discuss that. I can We can discuss ubiquitin. I think we discussed in the second session ubiquitin mediated mechanism. Yeah. So the ubiquitin mediated mechanism is like this: that how the ubiquitin is tagged onto the protein molecule, any of the protein molecule, okay, or any of the amino acid residues. So there are three enzymes which are part of this ubiquitin mediated mechanism. First is like E1, E2, and E3. So E1 is a ubiquitin 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 tagging enzyme, okay, which tags the ubiquitin. Uh, I mean, it transfers the ubiquitin to that enzyme E1. So specifically name, just a second. Uh, yeah, so this E1 is a ubiquitin activating enzyme, okay. E1 is known as ubiquitin activating enzyme. E2 is known as ubiquitin conjugating enzyme, and E3 is ubiquitin ligase. So this ATP, ubiquitin, in with ATP uh, binds to this act on this ubiquitin activating enzyme, and with the help of ATP, it will bind this ubiquitin activating enzyme will bind the ubiquitin molecule, and pyrophosphate and AMP adenosine monophosphate will be released. Okay, and after that, this E1 enzyme will transfer this E1 to the E2. Okay, transfer this ubiquitin to the E2 enzyme, and this E2 enzyme will transfer to the E3 enzyme, which is a conjugating enzyme. Okay, so since it is E3 enzyme, will conjugate the ubiquitin to the target protein. So this is how the system, I mean, the ubiquitin is transferred by the three enzyme complex. Okay, and after that, when the ubiquitin is tagged, there also is a so this ubiquitin tagged protein is then targeted to the proteasome system, onto which this protein is transferred onto this proteasome system, and it is degraded into a single amino acid residue, okay, chopped into a simple protein, uh, degraded protein. And further, when you if this ubiquitin mediated process can also be reversed by an enzyme known as deubiquitinase, okay, which removes this ubiquitin from the protein molecule, and then this protein molecule cannot be degraded. Okay, so to degrade the protein, ubiquitin is necessary, and this we talk regarding the ubiquitin, that proteasome system in the yeast generally there is a 26S proteasome system, and there are various subunits present. So here you can see this is a dimeric form of this enzyme. So this blue portion, you can see it is a regulatory subunit which recognizes the ubiquitinated protein at the top. Okay, and after that, AAA ATPase module is there. The pink portion you can see. So this AAA ATPase, what it does, it uses the energy from the ATP and helps to translocate the ubiquitinated protein onto the core particle, the yellow portion which you can see. Sorry, the orange portion what you can see. Okay, the core particle. It transfers to that core particle, and there inside the core particles the protein will get degraded. Okay, so it helps to move the protein ubiquitinated protein to the core particle by the help of the ATP. Okay, it has an inherited ATPase enzyme. So ATP will be converted into pyrophosphate, and that ATP energy is used to translocate it into a core particle, okay, inside it, and there it will be degraded. So this is how the proteasome mechanism is done. And generally, the in the ubiquitin as well, there are like this is a ubiquitin molecule, okay, and there is like lysine 48 and 63 uh, is helpful in forming the polyubiquitinated chain. So in the protein, generally there is a chain of polyubiquitination occurring. Okay, multiple ubiquitination occurs onto the protein. Not a single ubiquitin is there, but multiple. So that multiple ubiquitin are bound by this residues such as lysine 48 and lysine 63, which helps in the attachment of ubiquitin while forming a polyubiquitinated chain. Here you can see ubiquitin 1, ubiquitin 2, and it attached by the lysine 48 from the C terminus. Okay. Over here you can see lysine 63 attachment. So this residues 48 and 63 is important to form a polyubiquitinated chain, and this core particle is important. This whole core particle is called 26 size proteasome system. Okay, 26 size proteasome system it is called. So this is the mechanism of how the ubiquitination occur. And ubiquitination, for example, what is the ubiquitination is helpful in degradation of this cyclin and all, as we discussed earlier. The degradation, attachment of ubiquitinated chain onto the cyclin helps in the degradation process by the proteasome system, and then it regulates the cell cycle process over there. Okay, so maintenance, degradation, and activation of cyclin is important to activate the cell cycle. So that is the process of ubiquity annihilation. And apart from that, ubiquity annihilation is also helpful in targeting the MHC molecule. Okay, targeting protein to the MHC molecule. So antigen presentation cells, you know that there are many antigen presenting cells, immune cells, which are antigen presenting. So foreign DNA molecule, okay, which is, for example, bacterial DNA molecule, engulfed by the cells first, okay, and it recognizes the foreign. So it will then tag the ubiquitin to it, and it will degrade that protein molecule, okay. And after degradation, that protein molecule will be converted into small peptides, okay, and that small peptides will be translocated onto the MHC1 molecule on the mitochondria, okay. That MHC1 molecule will then be translocated onto the cell surface, okay, which acts as an antigen presentation to the other cells such as CD8 T cells. So CD8 T cells will recognize this peptide bound with the MHC1 molecule, and then it will trigger the inflammatory response by activation of other immune system, okay. So that is how the antigen presentation also occurs by the ubiquitin mediated process by degradation and presentation on the MHC1 molecule. So over there, it is also helpful. And in case of like glycine modification, for example, in histone. So as we talked in histone, that in histone generally the main modification which was occurring was methylation, acetylation, phosphorylation. Okay, and in some cases citrullination was occurring, but very few. Okay, as such in like third position of this arginine in H4, and also at second position of arginine in H3. Apart from there was no citrullination. Major was acetylation, methylation, and phosphorylation. And we talked about ubiquitin annihilation as well, that ubiquitin annihilation was present only in the H2A and H2B C terminal, okay, not in the H4 and H3. So this H3 in the H2A also, like third residue from the C terminal, ubiquitin annihilation or lysine residues is there. So when you want to, if there is a need for degradation of this molecule H2 and H2B, there it will get ubiquitin annihilated, okay, by the mechanism as we discussed, E1, E2, E3, ubiquitin ligating and activating enzyme, and then it can be degraded by the proteasome system, okay. And only, as you can see, only one site of ubiquitination is there in the lysine. Same in case of lysine, H2B also the third position of lysine at C terminal. So it is not a major modification that is present. Sumoylation is not there in the histone, okay. There is no sumoylation in the histone modification. Only this Y modification is there. But the ubiquitin mechanism is also very less prominent found in the histone. Is it clear?
Yes, sir. Thank you so much.
Yeah. So in today class, we will be discussing about the as you if you've gone through the week content, then in this week there was a class regarding the lecture was regarding the apoptosis and cancer cells. Okay, the characteristics of cancer cells and how cancer cells is produced promoted. And what are the different factors which promotes the cancer cell? Okay, and we were the discussion regarding the proto-oncogenes as well as tumor suppression genes, the genes which are involved in promotion of cancer cells were discussed, and the tumor microenvironment, what are the certain stimulants which promotes the cancer cell, that was discussed. And also there was a discussion regarding the apoptosis, the mechanism of apoptosis, and what are the total genes which are involved and how it is regulated. And specific discussion was on the neuronal cells, that how in the neuron cells, generally in the neuron cells, the cells are not dividing. Okay, so it divides at the initial stage of the development, and after that, the cells does not divide. So it goes on to the senescent phase. So in that case, generally the apoptosis is not an ideal condition to be generated in the neurons, because if the apoptosis is occurring, then it can be a harmful thing to for the organism, because the cells are not dividing, so it will degrade the cells over there. And apoptosis is not a harmful condition. Okay, in certain stages of development, uh, it is a beneficial process. But in certain condition, if it is deregulated, then it can be a harmful process as well. So we will see. If you have gone through the weak content, then we will revise. Okay, we will quickly go through what are the steps which are involved in the apoptosis, and what are the certain molecules which are involved over there. Okay, and how the regulation of apoptosis uh, is done inside the inside the cellular system. So apoptosis is an activation of the pre-existing genetic program that results in the cell death. Okay, and it is evolutionary conserved. It is a pre-existing genetic program that it is already present inside the genome. Okay, it is already governed that when the apoptosis should be occurring inside the cells. Okay, and it is not. It is the morphological features are like cell shrinkage, nuclear condensation, membrane blebbing, DNA fragmentation. Okay, so cell will get shrink, the DNA will get condensed, membrane blebbing will occur. As the membrane will start to uh, uh, form a bleb kind of structure, and DNA fragmentation also occurs. And cell death process requires ATP and in some cases protein synthesis. Okay, so certain proteins are required, as we'll be discussing, and also it requires ATP. Proteins such as apoptotic proteins, transcription and translation of apoptotic proteins are required for the development of apoptosis inside the cell. So then there are two different process of the cell death. One is necrosis and another is apoptosis, and both of them differs by each other. Okay, so in necrosis, there is a cell swelling, there is membrane rupture, organelle disarray, and inflammation. In necrosis, generally you find the inflammation process when the cells are infected by certain virus or bacteria. It start to it start to it start to degrade the cells. Okay, or certain immune cells which are infected by virus, it releases certain cytokines or other pro-inflammatory molecules that will lead to the death of the cells. Okay, of the nearby cells. That is called necrosis process, in which inflammation is a major part uh, occurring in that cell, and also organelles are ruptured in that rupture. Okay, organelles and membranes are ruptured. Whereas in apoptosis, the cells get shrink. Okay, it loses its volume. The volume gets shrink, and the organelles are preserved in that case. In apoptosis, the organelles are not damaged. Membrane blebbing occurs. Chromatin are condensed, and the dying cell get phagocytosed. So in apoptosis, the membrane gets flapped, the volume get reduced, and the membrane which is separated from this apoptotic cell are phagocytosed by the phagocytic cells. Okay, and it does not promote the inflammation. So these two differences important. That necrosis, it promotes inflammation. In apoptosis, you don't get inflammation. So as I told, that apoptosis is an important process during the development, and how it is helpful. It helps in the sculpting process. So one common example which we found in the apoptosis is the development of tadpole tails. Okay, the removal of tadpole tails during the development stage. So at the initial stage of the development, there is a tail present in the tadpole, but when it is converted into the frog, which is a mature stage of the development, the tail gets removed uh, from the organism. So that is at that stage apoptosis process is occurring. Okay, that deleting unwanted structures. So that is the best example for that. In the certain animals, certain organisms, at the hands. Okay, when the fingers, okay, hands of fingers of the hands. Okay, there are web kind of structure. In the hands, there are web kind of structure. If I just so this I'm showing the fingers, and there are webbing occurring in that around the fingers. Okay, around at the uh, s- connecting the two fingers. There are webbing at the initial stage of the development. So this webbing actually helps in the movement and other processes at the initial stage of development. But at the later stage, when the organism is mature, this webbing is removed. Okay, this webbing are removed by process called by apoptosis process, and this is an example of sculpting. So this helps in the sculpting of the fingers in the organism during the later stage of development. Third example is controlling the cell numbers. Cell number, if it is are too many, okay. For example, in the condition of the cancer, okay, during the suppression of the cancer, generally apoptosis process occurs that to limit the cell number. And if there is a deregulated apoptosis, okay, if the apoptosis machinery mechanism is not active inside the cell, it leads to the uncontrollable proliferation of cell, and that is the main reason for development of cancer in the organism. Okay, so apoptosis process is helpful, and certain chemotherapeutic drugs and certain other anti-cancer drugs also target the apoptosis mechanism to to limit the growth of the cancer cell. Okay, eliminating misplaced cells, certain cells which are disarranged during the developmental stage are also eliminated to that space. So when you study about the developmental stage of the organism, okay, during the initial stage of development, there are various cells modification which are occurring from the ectoderm to endoderm to mesoderm. There are various movement of cells during the development, and at that stage, certain cells are removed from that uh, portion, and certain cells are incorporated. So during that phase as well, apoptosis mechanism is found in the organism. So these are the four examples during the development in which the apoptosis machinery is applicable. And also during after the development, to maintain the homeostasis as well as we talk, to eliminate damage, harmful and abnormal cells. Okay, that apoptosis mechanism is essential uh, when the cells are infected with virus, or if there is a cancerous cells has a uh, which has a proliferative ability, okay, uncontrolled proliferative ability. During that stage as well, if the apoptosis machinery is active, then it can limit the growth of uncontrolled cell division, okay, and it can remove the harmful cells. So, apoptosis is not a damaging mechanism, but a helpful mechanism to the cells. So apoptosis in a pathological situation, if we talk, okay, so if too much apoptosis is there, that also can be should be regulated. Apoptosis should be regulated, as is case with all the process. Okay, if too much is there, then there are different diseases which occur, such as stroke, spinal cord injury, okay, neurodegenerative disease such as Alzheimer's and Parkinson's. So Alzheimer's and Parkinson's, there are accumulation of aggregated proteins inside the brain. Okay, aggregated proteins are there, and that are generally cleared by the apoptosis mechanism. And if there is too many apoptosis which is occurring, uh, because of the modified protein presence inside the cell, then the neurons will get degenerated. Okay, neurons will die because of the apoptosis. And as we talked that neurons are non-dividing cells. Neuron does not divide. Okay, so by apoptosis, if the neurons are getting degraded, then it leads to the disease such as Alzheimer's and Parkinson's. Okay, so there should be a very strict control on the apoptosis mechanism in the neurons. And also in the acquired immunodeficiency syndromes, AIDS, there is also an apoptosis, uncontrolled apoptosis is there, due to which the disease occurs. Too little cancer is the best example, okay, in which the apoptosis, due to deficiency in the apoptosis mechanism, cancer gets the ability to uh, promote the cell division continuously, okay, uncontrolled cell proliferation occurs. Also in the autoimmune disease, because in the autoimmune disease, generally the apoptosis mechanism is inhibited. In autoimmune disease, basically the self self stimulatory molecules, okay, are present, which act as an infective agents to promote the autoimmune disease. And generally, the role of apoptosis is to limit this condition, to limit uh, the molecule which is self damaging to the cells. Okay. For example, if you study about autoimmune disease, then there is rheumatoid arthritis. Okay. In rheumatoid arthritis, you have such antibodies. Okay, specific mechanism. We will not go into specific mechanism, but there is a self mono antibodies which reacts. Okay, it reacts with each other and it promotes the disease progression. And if there is a damage to the apoptosis mechanism, so in such case, the apoptosis will kill that cell. Okay, if it is self-reacting with the if it is reacting with the self antigen. Okay, but if it is not able to clear, then we can the autoimmune disease are generally present in that individual. Okay, so these are the uh, two examples over there. And generally, the proteins which are present in the apoptosis are in C. elegans and mammals. The process is same, but there is a difference in the proteins which are present. So in C. elegans, generally CED4, CED3, CED4 and CD3 is present, and the CED3 mediates the apoptosis process. Okay, and the CED9 actually inhibits the CED4 and CED3 to limit the apoptosis. So when we talk about the uh, there are FF1 caspase. Okay, so CED4 corresponds to FF1 and CED3 corresponds to caspases. And caspase is actually the proteins which promotes the apoptosis, which helps in activating the apoptosis pathway. Okay, other players are also involved, which regulates this caspase. So FF1 is one such factor, and FF1 is also regulated by cytochrome C, which we'll be discussing further. Okay, so FF1 will target the caspase, and then caspase will activate the apoptosis. And BCL2 family inhibits this apoptosis pathway, as was seen with the CED9. So these are the homologs of these C. elegans, which are found in the mammals. The role of caspase is we discussed in the apoptosis that it leads to chromatin condensation, DNA fragmentation, membrane blebbing, cytoskeletal changes, as well as phagocytosis. So this were the process which occurs in apoptosis is mediated through caspase. So if we talk about caspase now, then caspase are the mammalian homologs of CED3 gene, okay, which is found in the C. elegans. It is a homolog of CED3 gene. and it is a cysteine protease that cleaves after aspartic acid residues. So aspartic acid residues on the target protein. Okay, it is a cysteine protease. Okay, it will mediate proteolysis through cysteine residues, and it will cleave after the aspartic acid residues on the target protein on which it acts. Okay, so that is the mechanism of this caspase. And once activated, caspases cleave specific cellular proteins and induce the rapid apoptosis. So this is the role, and at least 12 known caspases is there, and we will discuss in further that what are the main caspases which are present. Okay, so here you can see there are over here you can see 7 total caspases, uh, initiator caspase as well as executioner caspase. Initiator caspase means the caspases which helps to activate the apoptosis process. Okay, which act as a act at the initial stage of the apoptosis mechanism. We will see in detail the signaling pathway, how it is occurring. But these are the initiator caspases which are present. Here you can see two domains which are present, domain S and domain L, small and large domain basically. And there are another domain which is present. One is card domain, which is present in caspase 2 and caspase 9. And in caspase 8 and 10, there is a DED domain, okay, death effector domain. This domain is actually helpful in binding, okay, in binding to its target protein. So caspase 8 actually binds with the receptor present on the cell surface, fast receptor, okay, by through this DED domain. So DED domain present on that receptor as well as DED domain present on this caspase will recognize each other and helps in the recruitment of this caspase 8 to that receptor. Okay, so that is why this domain is present, important in this caspase. And also in the caspase 9 as well, the caspase 9 binds to the FF1, okay, during the intrinsic activation of the apoptosis. It binds to the FF1. And in FF1, there is a card domain which binds to the card domain of the caspase 9 and further leads to its activation, cleavage and activation. Okay. So caspase will get activated after binding to the adaptor proteins. And after that, caspase will get activated and it will lead to the activity on the target protein. Okay. So this is initiator caspase, and in the executioner caspase, there is caspase 3, 6, and 7. In the initiator caspase, generally caspase 9 and caspase 8 are studied well. Okay. But in execution caspase, caspase 3 plays an important role. Okay, apart from other caspase. So here you can see caspase 3 activity. Caspase 3 generally activated by caspase 9 and caspase 8 through different mechanism. As we will be seeing, there are two mechanism. Okay, intrinsic mechanism of apoptosis. That means inside the cellular system, if there is a DNA damage or cellular stress or certain abnormal condition inside the cell, then intrinsic apoptosis pathway are activated. But if there is a certain external signals which are occurring, okay, which are present outside in the cytoplasm, outside environment, that also can be sensed by the cell to trigger the apoptosis by certain receptors present on the cells. Okay, so extrinsic as well as intrinsic mechanism, both the mechanism occur. And the final activation is of caspase 3. Caspase 3 is the final execution of caspase which is activated, and after that it leads to the apoptosis pathway through two different ways. So for DNA fragmentation, it activates this caspase activated DNase. Okay, so this CAD, that is a DNase, caspase activated DNase, is inhibited by this ICAD, inhibitor of caspase activated DNase. Okay, so when caspase 3 is activated, it acts on this ICAD and cleaves the ICAD and degrades it. Okay, so now this DNase is free. And the role of DNA is, as you know, that it leads to the DNA fragmentation. Okay, so in normal condition, this should not be activated. Otherwise, it will also lead to DNA fragmentation in the normal cell as well. So only when the ICAD is degraded, which is through caspase 3, then only the CAD will be active. Okay, uh, and this leads to the DNA fragmentation. Another mechanism is through this activation of ROC1, that is a row activated serine threonine kinase. So initially it is inactive, but by the caspase activity, it will become active. And how it is getting active? By caspase 3, as I told, that it leads to the cleavage after the aspartic acid residue. So it acts on the cysteine molecule after the aspartic acid residues, okay, on the ROC1. And this active ROC1 will now lead to the phosphorylation of this myosin regulatory light chain, MLC protein. And this MLC protein will then, phosphorylate MLC protein will act by a certain mechanism to induce a membrane blabbing. Okay. So myosin is a filament, intermediary filament, and this will lead to the membrane blabbing process. So DNA fragmentation, membrane blabbing was a two physiological consequence of apoptosis pathway, and that is done through two different mechanisms, the activation of caspase 3. So this is the mechanism through this is an extrinsic apoptosis pathway through which the caspase 3 is activated and leads to this apoptosis function. So as I told that the extrinsic system are the stimulant or certain stressors which are present in the extracellular in the extracellular region of the cell, okay, is recognized by the receptor present on the cell surface, that is FAS FAS receptor is present as well as TNF receptor is present. Okay, so this TNF as well as FAS receptor will recognize these stressors, and it will recruit the caspase 8. This caspase 8 will bind to the FAD region. Okay, FAS associated dead domain, that is called. So FAS associated dead domain, it binds to the caspase 8 through dead effector domain, as we discussed over here. Okay, dead effector domain. So both these proteins shares the dead effector domain, and this helps the caspase to recruit to that site. And after binding and activation of caspase, it leads to the activation of caspase 3. Okay, and caspase activation and also the other caspase, which leads to the phagocytosis process. But main role is of caspase 3, which leads to the chromatin condensation, DNA fragmentation, as well as membrane blabbing changes by activation of uh, MLC protein as well as the CAD. Okay, caspase associated DNAs. So this is regarding the extrinsic pathway. Now, if we talk about intrinsic pathway of apoptosis, so in intrinsic pathway, generally the DNA damage or certain stress present inside the cells or other stimulus which which are toxic to the cells are recognized by the intrinsic protein present. Here over here receptor is not present. Okay, but the intrinsic mechanism helps to regulate the apoptosis mechanism. So this stress or other stimuli or DNA damage uh, work on this BCL2 family. Okay, BCL2 family generally helps to limit the apoptosis process. Okay, as we discussed earlier, that BCL2 is an inhibitor of apoptosis. In BCL2 family as well, there are many other different proteins. But in general, here we are talking that BCL2 family inhibit apoptosis. So if you want to promote the apoptosis, then this stress or other stimuli will inhibit this BCL2 family. And if you inhibit this BCL2 family, then by the apoptosis will get promoted. Okay, so there is a cytochrome C, which is present in the electron transport chain. Okay, ETC, during a generation, which is helpful in the generation of ATP molecule. Cytochrome C is released from the mitochondria into the cytoplasm. Okay, translocated from the mitochondria to the cytoplasm. Over there also there are certain proteins. Okay, bead bags are there. We'll be discussing it further at how the cytochrome C is getting released. So cytochrome C released by this mitochondria acts on this FF1. Okay, It binds to this FF1 and activates the FF1. And FF1 in turn binds to Caspase-9 through card domain. In Caspase-9 we discussed there was a card domain, and this card domain is also present in the FF1. Okay, so there is a linkage of the Caspase-9 with FF1 through card domain, and due to that the Caspase-9 gets activated. Earlier it was pro-caspase, now it is converted into a caspase, active caspase molecule. This caspase-9 activates the caspase-3, and this caspase-3 further leads to the same function as we discussed through extrinsic pathway, okay, chromatin condensation and membrane cleavage. So both this process, extrinsic as well as intrinsic, are independent of each other, okay. It is not a dependent pathway, both are independent, but the end molecule activation is the same, that is caspase-3 activation, and also the other pathways which plays a role. So here you can see the summarized version, caspase-8 activation by the Cas ligand and then caspase-3, and also the intrinsic process pathway. Through this pathway, the caspase-3 is activated. So the formation of this caspase-9 FF1 cytochrome C complex, it is called apoptosome formation. Okay, this complex is called apoptosome. Complex of caspase-9 FF1 with cytochrome C is called apoptosome. This apoptosome actually activates the caspase-3 molecule. Is it clear to everyone? This clear?
Yes, sir.
Yeah. So, so now, as we talk about BCL2, that BCL2 is a BCL2 family generally inhibits the apoptosis pathway. But in that case, also in BCL2, it is not an anti-apoptotic only. There are also pro-apoptotic uh, family as well. Okay, so the BCL2 family in general contains three kinds of different kinds of protein. One is anti-apoptotic, another is pro-apoptotic, and third one is BH3 only. So we will discuss what is the role of this BH3. Okay, further. But anti-apoptotic includes BCL2, BCLXL, BCLW, A1, MCL1, in which BCL2 and BCLXL are two main BCL, okay, pro anti-apoptotic molecule which are found and which is regulated. So, and in pro-apoptotic, generally PAC and BOC is there, okay, present. PAX and BAC are the main anti-pro-apoptotic molecule which is present in which act as a pro-apoptotic molecule. And here you can see the transmembrane domain is there in both the case, and in the and BH2, BH1, BH2, and BH3, all the all the three domains are present in anti-apoptotic as well as pro-apoptotic. In anti-apoptotic, BH4 is extra, okay, and in BH3 only family, only the BH3 domain is present, and in BH3 also there is a bead, beam, big, bad, Noxa, Puma. Okay, so all this BH3 family actually helps to promote the apoptosis pathway by targeting, by inhibiting the anti-apoptotic protein and activating the pro-apoptotic molecule. Okay, so that is the role of BH3. So here we are seeing that here is a view of the how this BCL2 family act with each other to regulate the apoptosis. Okay, so as we talk that DNA damage, stress, or other stimuli, as well as growth factor withdrawal. Okay, so as you know that energy is important to the cell, and if you withdraw the growth factors from the surrounding, that cell will not be able to survive in that environment. Okay, and we'll also see in this lecture as well as we had discussed earlier as well, that sorry, that growth factor actually helps to promote the cell cycle. Okay, and cell cycle progression occurs uh, by activation of cyclin complex. Okay, by transcription of this. cyclin D by E2F growth factor. We had discussed earlier that. Okay, and promotion, the division of the cell. So if you withdraw the growth factor, then cell will not be able to grow, and in that condition, generally the process of apoptosis is found. Or apoptosis is generally occurs in that cell. So BH3, these factors generally acts on this BH3 only protein first, not directly onto the BCL2. Okay, the apoptotic protein. It acts on this BH3 only protein first. I mean, it sends these factors are sensed by this BH3 only protein, such as bead, bat, noxa, beak, beam. Okay, and this inhibits this BCLXL and BCL2 as well as activate the backs and back. Okay, so if you want to promote the apoptosis, then you need to inhibit this BCLXL and BCL2 and activate backs and back. Okay, so this individual protein plays a role in doing that, and when if the backs and backs is activated, then the back and backs actually do the holes in the mitochondria. Okay, creates a pore in the mitochondria, and through that pore, the cytochrome C is released outside the mitochondria. Okay, so the role of back and backs is to form a pore on the membrane of the mitochondria, so that the cytochrome C will get released. And cytochrome C, as we discussed, it will act on this APAF1, which further binds to caspase 9. Caspase 3 activation occurs, and then the apoptosis formation. So this is the total regulation. This is how the BCL2 family helps in the regulation of apoptosis by sensing and activating the caspase molecule. Now, apart from caspase, there are also P53 activation. Okay, so this P53, as we discussed, that it is a tumor suppressor gene. We discussed earlier as well. Okay, about this P53, how it was getting activated and how the activation of P53 leads to the suppression of the cell division. Okay, it is a tumor suppressor gene, and the P53 generally activated by during the DNA damage process, or if there is a single-stranded DNA break, single-stranded or double-stranded DNA break. At that time, P53 is activated by proteins such as ATM and ATR, if you remember. Okay, so this P53 uh, promotes the transcription if there is a persistent DNA damage. Okay, at the initial stage, P53 will not directly activate the apoptosis mechanism, but if there is a persistent DNA damage which is beyond repair for the cellular system, then that cell does not have any option but to undergo the apoptosis process. Okay, so at that stage, this P53, active P53, beyond the actual concentration, okay, will activate the Noxa Puma, which is a BH3 protein, BCL2 family member, and also the transcriptional activation of PAX. P53, as we discussed, it is a transcription factor. Okay, so it will transcriptionally activate this BH3 family protein, BH3 family, as well as the proapoptotic molecule. And this protein, as we discussed, that it will promote the apoptosis by releasing the cytochrome C. Also, P53 directly activate the release of cytochrome C by forming a pore. Okay, by directly interacting with the mitochondria and activate with the signaling mechanism and release the cytochrome C. So through both this pathway, P53 act and it leads to the formation of apoptosis during the persistent DNA damage. Okay, so and generally in the cancer, so we discussed that in cancer the apoptosis mechanism is inhibited. Okay, because of the limited apoptosis or no apoptosis, the cancer acquires the ability to uncontrolled proliferation. Okay, So how? Because P53, as we discussed, it is a tumor suppressor gene, and if there is a defect in the P53 such that it does not activate the DNA apoptosis pathway, it will not. If there is a mutation in the P53 or there is a defect in the activity in P53, it will not lead to the activation of this apoptosis pathway. Okay, and that is generally found in the cancer cell. Mutation in the P53 that will limit the apoptosis, and it will also does not sense the DNA damage as well. Okay, if the P53 is mutated, so that is why it is called tumor suppressor gene. Tumor suppressor gene means to suppress the tumor. So P53 by activating the apoptosis pathway suppress the tumor. That is why it is called P53. Another class of genes which is present in the cancer cell is oncogene. Okay, so oncogene is a gene which the activation of which promotes the tumor. Okay, so for example, certain genes, certain transcription factor which leads to the uncontrolled activation of cyclins. For example, it's just an example. And okay, if the activity of this protein is there, then it will lead to uncontrolled division. Okay, for example, uncontrolled activation of cyclins. And cyclin, you know that it will lead to the cell cycle activation. Okay, by binding to the CDKs. And in case other proteins as well, the transcription factor which leads to the transcription of certain proteins, which helps in the uncontrolled division or uncontrolled proliferation of that cell. That is called oncogene. Okay, so tumor suppressor gene, when activated, it will lead to the inhibition of cancer. Oncogene, when it is repressed, it will lead to the inhibition of cancer. Okay, so these are the two difference. Okay. So now we will discuss that what are the mechanism which can be regulated on the caspase 9 and 3. Okay, so here there was a regulation that we are talking about the regulation over here, that how apoptosis can be regulated first. So this at the initial stage of regulation after the before the release of cytochrome C. Just a second. At the initial stage of the apoptosis, generally the caspase, the BCL2 family, this anti-apoptotic protein regulate inhibit these backs and back. Okay, before release of cytochrome C. The regulation also occurs after the release of cytochrome C, the inhibition of the apoptosis. Okay, so there is a protein called IAPs, inhibitor of apoptosis proteins. Okay, so this protein inhibits this caspase 9 activation of caspase 3, and also it inhibits the caspase 3 activation of apoptosis pathway. Okay, so if certain cells which does not follow the apoptosis, for example, neuronal cells. Okay, so neuronal cell generally this protein is present in higher number, inhibitor of apoptosis protein, because the neuron cells will not undergo apoptosis. Okay, so it regulates the caspase 9 activation of caspase 3. It inhibits the caspase 9 activation of caspase 3 as well as caspase 3 activation of apoptosis. And also this protein is also in turn regulated by another protein which is released by the mitochondria, such as SMAC. Okay, so if there is a DNA damage and if there is a release of cytochrome C, and if you want to promote the apoptosis, then you need to also target this and degrade this protein as well. Okay, so that the inhibition will get repressed. Okay, it will get loosened, the inhibition. So mitochondria, in addition to cytochrome C, also release SMAC protein, and this SMAC actually inhibits this protein IAPs, so that it will lead to the activation of caspase 9 it will lead to the activation of caspase 3 and apoptosis pathway. Okay, so in neurons generally this protein is found in higher number and the SMAC level is very low in the neuronal mitochondria. Okay, in the neuronal mitochondria, SMAC level is very low, so that it will not able to inhibit this protein. And even though the cytochrome C is released inside the neuron, okay, the cytochrome C will not be able to trigger the apoptosis pathway in certain cases. Okay, in certain cases, it is not like in neuron, as we'll be discussing further, that in neuron, at some part, the apoptosis mechanism is essential, but in the most of the cases, the apoptosis machinery is not essential. Okay, so even the cytochrome C is released due to apoptotic signal or other mechanism as well, there need to be another regulation. Okay, so that uncontrolled apoptosis does not occur inside the neuron. So by this way, the cells can limit the apoptosis pathway. So as we discussed that in the neuronal cells, there will be stricter controls so that apoptosis does not occur randomly. So first thing is to maximize the anti-apoptotic checkpoint. Okay, the anti-apoptotic checkpoints is over here in the neuron. Additional checkpoint is present, such as IAP, which inhibits this caspase-9 activation of caspase-3 and apoptosis pathway. And second thing neuron cells do is that it minimizes the pro-apoptotic checkpoint. Okay, so in neuron, in general, backs and back both are found, but in neurons generally only backs is found. Okay, not back. So in this case, the only backs is there. So even though cytochrome C will be released, but in very less quantity. Okay, because backs is a little less activator of cytochrome C. Back generally leads to higher activation of cytochrome C. So this is the another regulation which is found in the neuronal cell to limit the apoptosis. So this is the overall summary that how the regulation occurs in the regulation of apoptosis occurred in the neuron cells. One is the IAP regulation, another is the BAX inhibition. Okay. So there is a question that why do mature neurons not completely shut down the apoptosis pathway? Answer is that neuron utilizes many components of the apoptosis machinery, such as BAX and caspases, for synapses and axon pruning. Okay. So during the even though the neurons does not die, okay, does not get apoptosized or division does not occur, the neuronal cells remain stagnant, but the neuronal connections which is occurring, the synapses which is occurring by one neuron to another. Okay, so you already know neuronal modeling. Okay, the plasticity in the neurons. If you have gone through that term, there is a plasticity to that neuron. So during the plasticity process, there is a various different connections which are occurring between the neurons. Okay, and various different synapses occurs between the two neurons, and also the axons pruning occurs. Certain part of the axons are removed, certain parts are added during the neuronal connections between the two neurons. At that stage, generally, by the help of these backs and caspases, by using the apoptosis machinery, only that connections are made. Okay, so over there, it is required. Apoptosis is required, but to a limited extent. Okay, so only some portion of these proteins are activated at certain stage and at the certain part of the neurons, not the whole neuron. Okay, so this was regarding the apoptosis. So if you have any doubt in the apoptosis process, uh, you can ask. Is it clear to everyone? The apoptosis machinery mechanism?
Yes, sir. Yes, sir.
Okay, thank you very much. So now we'll be talking about cancer cells. Okay, so if you permit, then we can take like 5 minutes break. Okay, 4 to 5 minutes break, then we can continue. let's take a break of 5 minutes. Yeah. So, is it audible?
Yes, sir.
We can continue. Okay. So, now let's talk about cancer cell. Okay. So, in general, you all have the idea about how cancer, what is cancer. Okay. Cancer is an uncontrolled cell division, and when the normal regulation of the cell division is dysregulated, then there is a cancer development. Okay, that is uncontrolled cell proliferation. Key characteristics: unrestrained proliferation of the cells, and it invades to the surrounding tissues and spreads to the other organs. So these are the two main characteristics of the cancer cell. So there are two kinds of tumors basically. One is benign, another is malignant. Okay, tumor is basically the abnormal growth of the cell, that is called tumor. Benign means the remain restricted to the original location, that is non-invasive. The cancer which are, any query?
Sir, your voice is echoing since from the time you have put your laptop for charging. Since then, it is echoing.
Mm, but without charging, like it will shut down.
Now it is good, sir.
Now it is good.
Yes, yes.
So just cross check again whether it is the charging. Is it good or is it echoing?
It's perfect now.
Okay, okay. Because without charging it will shut down. So yeah, so malignant means the invasive property of the tumors. Okay, and this is the stage at which the cancer becomes uh, dangerous to the humans. Okay, the metastasis stage of the cancer. So translocation from the site of the generation to the other site or the location in the organs. That is called metastasis, and that at that stage generally it is difficult to target. Okay, target the cancer cell. There are different types of cancer, such as carcinomas, which arises from the epithelial cells. Okay, and that is the most common type of cancer from the epithelial cell which arise. Sarcoma arises from the connective tissues. Leukemias, which is cancers of the blood, okay, lymphocytes as well as hematopoietic cells, uncontrolled division of that cells. There's a cancer from the uncontrolled division of cartilage, that is chondrosarcoma, and also the melanoma, which is a pigment cells. So these are the five different types of cancer, and also the cancers arising from the cells of the nervous system, such as glioma, glial cells, okay, which are present in the nerves. The macrophage, which are present inside the nerves, that is called glial cells. Okay, and uncontrolled division of that is called from glioma. And also the neuroblastoma, which is cancer from the immature nerve cells. So development of cancer, as you know, it promote development occurs through somatic mutations. Okay, most cancer arises from a single cell. If there is a aberrant changes in the single cell such that it loses the general characteristics of the cells of regulation. Okay, the apoptosis pathway are the main target of the cancer cell. Okay, deregulated apoptosis pathway or deregulated sensing pathway. Okay, of the damage sensing pathway is deregulated, or certain genes or certain proteins are inactivated, or abnormal activation occurs of certain proteins. At that stage, there is a cancer development. and generally it arises from a certain subset of cells or from a single cell, and that single cell actually proliferate continuously to promote a subsets of cell. That subset where again then continuously compounds itself and then promotes a tumor cell, okay, which contains a very large number of cells, cancer cells. Somatic mutation is the main thing which occurs, okay, the mutation in the DNA of the cells, which provides resistance to certain mechanism, controlling mechanism inside the cell, which that is promoted by certain mutagenic agents, which enable development of cancer. Okay, mutagenic agents are many. There are many mutagenic agents which binds to the DNA molecule and leads to the change in the sequence. Okay, bases, purine and pyrimidine molecule, base pair. That mutagenic agents are in different. Many such agents are there, which leads to that development. And it is a multi-stage process. Accumulation of mutations occur continuously, and then it leads to the uh, development of the cancer. Okay, so here you can see different stages of the cancer, such as stage one, which is localized to certain part, certain area, okay, of the organs. In stage 1B, here you can see it is slightly started. It is proliferating in that region, occupying that region. Stage 2, here you can see it is translocating from that membrane to the muscular membrane. Okay, this is an example of the stomach. In stomach, first there is an outer lining, then there is a muscle, then the supporting tissue, and there is an inner lining. So in stage 2, here you can see it is translocating from the supportive tissue into the muscles. Stage 2. In stage 3, generally it is further moving towards the interstitial space. Okay, and stage 4, it is completely metastasized and promoted and is transmitted to the different organs. This single cell. So only one cell is required. Okay, if one cancerous cell is translocated to other organs, then it will differentiate itself into many cells, clonal propagation, and then it will lead to disease condition in that organ. So at this stage, stage 3, stage 4, it is very difficult to control the cancer patient, okay, and it's very difficult to target. At the initial stage, such as stage 1, stage 2, you can still use certain chemotherapeutic agents to limit the cancer cell growth, okay, by removing the cancer cells. And the targeting of cancer cells and the treatment of cancer cell is difficult in case is because if there is single cell which is surviving, okay, after the chemotherapeutic treatment and killing of the cancer cell, if there is a single cell which is surviving, then it will again lead to the promotion of the many cells after a certain period of time. So the treatment should be accurate that no cancer cell should be present. The treatment should be sensitive enough that each and every cancer cells are targeted accurately. So that is why it is very difficult to, in some cases, generally at the later stages, it is very difficult to uh, I mean, control the proliferation of the cancer cells. Uh, treatment treatment is very difficult at the later stage. So cancer cells also exhibit genomic instability and chromosome aberrations are there. Okay, and due to these are also part of the mutations only. Okay, certain chromosomal dysfunction which codes for the normal chromosome which codes for a normal gene, but there is a translocation. For example, in translocation, certain portion of the chromosomes are translocated to other chromosome, and then the genes which are encoded by that chromosome, okay, uh, becomes non-functional, or it will be irregularly activated. Okay, so irregular activation will be occurring. So that is how it promotes the cancer cell development. By at the genetic level, on the changes in the genetic level only constitute the cancer. Okay, Um, so this you can remember. Inversions, certain orientation of the genes are reversed so that it will that gene will now become inactive. Okay, because the orientation of the gene is reversed, as well as deletion of that gene. If there is any tumor suppressor gene, okay, and if that gene is deleted, let's take for example, P3 gene. If that is deleted, then now there is no regulatory mechanism, which is no regulatory protein which is present to control the apoptosis. So this is one of the mechanisms by which cancer cell is able to proliferate. Amplification of certain oncogenes, okay, certain genes which helps in the activation of tumor. The amplification of certain genes will lead to further protein production, abnormal protein production of that protein, okay, which further leads to the disease development. So these are the certain chromosomal arrangement and modification which occur in the chromosome due to which there is a development of cancer. And also in the cancer cell, there is an Warburg effect. There is a term called Warburg effect, in which the cancer cells generally, if you are aware, then cancer cells are the surrounding environment over the cancer cells are acidic in nature. Okay, it is a glucose deficient. Sorry, it is an oxygen deficient area. The cancer cells which are surviving. Okay, the oxygens are very limited over there, and there is an accumulation of lactate inside in the cancer cell environment. So this lactate actually promotes the acidic environment, and this is the characteristics of the growth of the cancer cell, and that is called Warburg effect. Okay, depletion of oxygen and promote production of lactate. How lactate is formed? If you are aware of the glycolysis pathway, then pyruvate is converted into lactate product. Okay, if there is an excess amount of pyruvate, then that pyruvate is converted into lactate. So how pyruvate will be formed in the cancer cells? Generally, the cancer cell, there is an abnormal accumulation of glucose. Cancer cell import large quantities of glucose inside, okay, inside the cells by the GLUT receptor present on the membrane. So the GLUT receptor will uptake the glucose molecule, and it will be large quantities. Certain part of this, and it will go through the glycolysis cycle and produce the pyruvate. Certain part of the pyruvate will be constituted, will go through this TCA cycle, and it will help to make the amino acids and other proteins which helps in the growth of the cancer cell and also the generation of ATP by the mitochondria. If there is an excess of pyruvate, then it will convert into lactate. Okay, and that lactate is actually accumulated inside the cells, inside the cancer cells. And the environment of cancer cells are acidic in nature due to lactate presence and due to abnormal accumulation of glucose. Okay, so this lactate will be translocated outside to the receptor present on the cells, and this lactate generally present. So this is also uh, one of the markers of the cancer cell, that if there is an higher amount of lactate present in the cancer cell surrounding, then uh, that 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 region, we can say that the cancers, it is a cancer cell producing region. Okay, if there is a higher amount of lactate which if you detect in the surrounding of these cells, so there is that region might be prone to the cancer development. This is also one of lactate is a marker of the cancer detection. Okay, so there is a shift from the oxidative phosphorylation, such a production of ATP, to the aerobic glycolysis. Okay, so glycolysis pathways are majorly active in the cancer cell. Tumor microenvironment also plays an important role in the cancer cell development, and certain immune cells such as fibroblasts, WBC, and certain other cells such as endothelial cells, smooth muscle cells, promotes the development of cancer, okay, by secreting certain factors or the from that cells or cancer cells secrete certain factors which helps in the growth and development of that immune cells. So the major thing the cancer cell will do over here is that it will suppress the immune response. Okay, immune response, if it is activated, then it will limit the cancer. It will degrade the cancer. So cancer cell will secrete certain factors which will modify the immune cells to limit the immune response to that cancer cell, and it will facilitate the growth of cancer instead. Okay, so it will make friends. Okay, so immune cells are modified by secretion of certain factors by the cancer cell, such that it helps to grow the cancer cell further by secreting. First, the cancer cell will secrete certain molecules which will change the immune cell. And after changing the immune cell will secrete certain growth factors, certain other factors which helps to promote the cancer cell, okay, promote the growth of cancer cell. That is how the crosstalk occurs between the immune cells and the cancer cell. And generally, the target over here, the therapeutic target over here is to first limit the cancer cell secretion of certain cytokines or certain factors which can modulate the change in the immune cell to cancer promoting. Okay, that is one target. Another target is to help this immune cell release certain factors which can limit the cancer cell growth. Okay, so for first thing is to not be able to change the immune cell. Okay, another thing is to secrete certain factors which limits the cancer cell growth. So these are two main therapeutic targets which generally which people do a lot of research in this. Okay, and it is an active research field in which instead of targeting directly the cancer cell, if we can modulate the immune system so that the immune system will target the cancer cell, that is a better therapeutic strategy. And you might have gone through various such tech, if you have learned through, okay, various therapeutic strategies are developed through that. One such recent recent therapeutic strategy is CAR T cell therapy. Okay, so in CAR T cell, what it does is that there is a chimeric antigenic receptor on the T cell. The role of T cell is to kill the to secrete certain cytokines or factors. Okay, so that factors will kill the cancerous cell. Okay, and CAR T cell is designed in such a way that there is a chimeric antigenic receptor. So there is a receptor which is chimeric in nature. Okay, so activation of that receptor will lead to the release of these cytokines. Okay, and these cytokines will actually kill this cancer cell. And there is an also regulation in that cell that even though the cancer cell will secrete certain, sorry, there is a regulation that even though the cancer cell will secrete certain factors, it will not change the physiology of that T cell. Okay, and that is how you can read about it for this class. You can read about CAR T cell therapy, and it is a very interesting technique to target the cancerous cell, okay, by T cell modulation of the receptor on the T cell. So you can read about it, detailed mechanism. Also, there are many other immune cells such as macrophages are there, and basophils, and all the other immune cells, like neutrophils. Okay, macrophages also plays an important role over here. There is fibroblast cells, which helps in the promotion of tumors. So all these cells helps in the modulation of these cells helps in the development of cancer. So here you can see the modulation. There is a release of TGF beta, VEGF, IL-1 beta, IL-6, TNF alpha, IL-10. So all these factors which are released by the cancer cell helps the modulation of this other immune cells, okay, and makes the immune cell adaptive to that cancer environment so that it will not be able to kill the cancer cell. So one such factor is this TGF beta, which converts this fibroblast into the cancer associated fibroblast. So now this fibroblast will not be able to kill the cancer cell, as well as it will further facilitate the growth of cancer cells. So cancer cell will secrete TGF beta and it will modulate the fibroblast. Another another example is the release of this IL-1 beta, IL-6, TNF alpha, which is a pro-inflammatory cytokines, okay, which makes the cancer cell convert the cancer cell from epithelial to mesenchymal, okay, mesenchymal. And this EMT transition is important for the metastasis process of the cancer. And as we discussed, that metastasis is a very harmful condition. Okay, if the cancer cells start metastasizing, then it is a very harmful condition and very difficult uh, stage to stop the cancer growth. Third stage is the angiogenesis. New blood vessels are formed because the cancer cells are dividing rapidly, so it will require more blood over there. So angiogenesis are higher in the cancer cell. Okay, abnormal vessel growth occurs, as well as immune evasion. Immune evasion is there. Okay, polarization of pro-tumor M2 macrophage and recruitment of pro-tumorigenic T regulatory cells. So monocytes monocytes is converted into macrophages, okay, by activation. And these macrophages are two kinds, M1 and M2. So M1 is a pro-inflammatory macrophage which targets the tumor. Okay, it targets the tumor and limits the growth of cancer. M1 macrophage, but by certain stimulant, if it is converted into M2, then that M2 macrophages release certain factors which helps in the growth of the tumor. Okay, so there are two different populations, two main populations of macrophage. Apart from that, there are also other populations of macrophages, but these are the two main populations. So cancer cells secrete certain factors which helps to promote the M2 macrophage and limit the M1 macrophage. Okay, so that it will lead to the growth of the tumor. And also the T regulatory cells. So the role of T regulatory cells is to inhibit this T cell. Okay, CD8 plus T cell. CD8 plus T cell is a killing cell. Okay, it is a apoptotic. It is a cytotoxic T cell which directly binds to the antigen present on the cancer cell and kills it. Okay, by releasing certain cytokines, granenzymes and cytokines. So T regulatory cells the cancer cell will secrete certain components which will activate the T regulatory cells. And T regulatory cells in turn release these factors such as TGF beta, IL-10, which inhibits the cytotoxic T cell. And if cytotoxic T cell is inhibited, then cancer cell will be able to survive. Okay, it will not degrade the cancer cell. So this is the mechanism by which the immune cells work. Okay, and the crosstalk between the cancer cells and the immune cells occur by this mechanism. The tumor microenvironment is a very complex interplay of various factors released by the cancer cell as well as by the immune cells, and very complex interplay occurs. Okay, and people are still there is an active research field completely devoted, okay, to study what are the different regulations and occurring, and many monoclonal antibodies are designed to target this tumor microenvironment. Okay, directly target the immune cells, which can lead to the deregulation of this cancer cell development. So many therapeutic strategies are there, but all these therapeutic strategies till date is not 100% foolproof because many different cells are involved and many factors are released, and various signaling pathways are activated as well as deactivated. So it's still an active area of research. So yeah, so now this tumor site, as we discussed, that the metastatic cancer cell is the main culprit, which lead to the uh, translocation of this cancer from one organ to the different organs. Okay, So in that also, stem cells are the main important cancer cell which is present. Okay, cancer stem cells. So the stem cells, this other population of the cancer cells, okay, are derived from the stem cells only. So initially there would be a stem cell which has an uncontrolled division ability, okay, uncontrolled proliferative ability, and that from that stem cell, other populations of cells are formed, and this other population does not have the metastatic capability, but only the stem cell will have the metastatic capability, okay. Stem cell, as you know, will metastasize, and if it translocate to other organs, then that stem cell have the property to make various clones of that single cell, and it will infect their organs, and it will lead to the cancer development in different organs. So targeting stem cells is the main thing over here instead of targeting the normal cell. Because if you target the normal cell and if only one stem cell is present, then again the after some period of time the condition will be the same, and you might be studying the you might have known gone uh, you might be knowing this terminology called relapsing. Okay, so after chemotherapeutic treatment, again the cancer got relapsed. Okay, and again the cancer got formed. Since that stage, actually, the his differentiated progenies got degraded, but they targeted the differentiated progeny, but the population of certain population of stem cells still remain active, and after some period of time, that from that stem cell, further cancer got promoted, and the relapse occurred in the patient. So, active research in this field, which specifically targets stem cell, okay, that is the therapeutic strategy which and the research area we need to focus on over here, okay, to limit this disease. So this is the I mean summarized view of the hallmarks of the cancer, okay. Certain what are the different conditions which are present inside the cancer cell, okay, which helps in the promotion of cancer cell and proliferation. Summarized view. So there will be genomic instability and mutation. Then enabling replicative immortality. So uncontrolled replication will be there in the cancer cell. Cell death will not occur because apoptosis pathway is inhibited. Metastasis property will be there in the cancer so that it will translocate to other organs. Angiogenesis will be higher in the cancer cell so that it will promote the blood vessel proliferation. Okay, blood vessel supply, the all the growing cells. Growth suppressors would be inhibited so that it will not lead to decrease in the cell proliferation. Proliferative signal will be activated. Higher proliferative signals will be there. Certain transcription factors are activated, which leads to the increased activation of the growth factors and proliferation. Cellular energetics will be deregulated. Higher number of ATP will be produced. Lactate glycolysis will be higher in number due to accumulation of glucose. Lactate will be higher in number, which will promote the acidic environment. So deregulated cellular energetics will be there. Avoiding immune destruction. The immune cells will be specifically trained that so that it will not be able to kill the cancer cell. And tumor promoting inflammation. Immune cell will be trained that it will lead to the further proliferation of immune cell by releasing certain factors. So these are all multiple things are accumulated to promote the cancer. Okay, it is not a single thing. Multiple factor occurs to promote and develop the cancer cell. Okay, and because of that, only there is this disease is very hard to. I mean, although the recent research and in the current field, people are still researching, and there is active Yeah. Any doubt? Okay, so this is the total accumulation of all the factors which promotes the cancer, and people are still studying and the mechanism and the various cells involved to design a therapeutic strategy. Although none of the strategies currently is foolproof. Okay, so we have like 15 minutes. Other things are there, but let's discuss some questions. Okay, which were present in this from this class as well as from the assignment, previous assignment. We will go through the question. I will just uh, show the answers and discuss what will be the correct answer. And if you have any doubt in the question, then you can directly tell over here. So first question, this is some model questions regarding the today's class. Okay, so this is about the senescence. We have not gone through the senescence and aging. We only talked about the apoptosis, but I will explain over here what is there in this question, and we will discuss the probable answers. Okay, so pick the correct statement about senescence. Senescent cells remain irreversibly arrested at Z0 phase. The second thing is senescence is an active cellular death process. In senescence is a stage process in which the growth of the cells, okay, the cell cycle is inhibited. The uh, cell uh, does not pass the G1 phase, okay, it is arrested in the G0 phase. It is irreversibly arrested at the G0 phase. There might be a certain various factors which are involved. One such factor is, for example, growth factors are deficient in the environment. Okay, or after a number of cell division, the cells become arrested in the Z0 phase due to telomere shortening. Okay, so telomere are the ends in the DNA molecule, and if the telomere gets shortened at that stage, the cells get arrested. Okay, the cells will not be able to replicate at that stage. Uh, and so at that stage, the cells undergo the senescent stage cells, ideally the dormant stage of the cells. Okay, so it is not a dead process. It is not a dead process, but then dormant stage. Okay, so cell death does not occur in that stage. Occurs only due to infection? No, it does not occur only due to infection. Basically, it occurs due to if there is a higher number of replication in that cell. Okay, up to a limited number of time. After a limited number of replication, the cell will undergo senescence. So it does not occur due to infection. Chromosome fragmentation and cell shrinkage are the main characteristics. Uh, Not ideally because the cell will not undergo death process, and in the apoptosis generally the chromosome fragmentation and cell shrinkage occurs, not in the senescence. Okay, in senescence the cell morphology remains ideal, but the replication and the cell division is inhibited. The correct answer is option one, that is senescent cell remain irreversibly arrested at C0 phase. The second option is which of the following is not a hallmark for aging. So in aging also, as we discussed, that due to aging only, due to senescence, the aging occurs. Okay, if the cell undergoes senescence, that is the one hallmark of aging. And in the senescence, as we discussed, the telomere attrition, that is telomere gets shortened at the ends. That is one consideration. Okay, for undergoing the aging. Another is the mitochondrial dysfunction. For example, the mitochondria will not produce enough ATP molecule. Okay, and it will not be functional to produce the cellular energetics. Okay, the mitochondria is the main hub to produce the energy. Okay, and where other cellular process. If it is dysfunctional, which which occurs in the during the aging process, mitochondria is dysfunction. Okay, so this is also one of the hallmark of aging. Stem cell exhaustion, yes, certain stem cells. So, for example, hematopoietic stem cells. Okay, from the hematopoietic stem cells, certain blood cells are originated. Okay, from the bone marrow. and also other stem cells from which the other different cells are originated. So if stem cell is exhausted, then that that progenitor cells or the cells which are derived from these progenitor cells will not be able to form. Okay, and that is one hallmark of aging. So these three is correct. Low ROS, no. Because of high ROS, ROS means reactive oxygen species. Okay, if there is a higher reactive oxygen species, then there is a higher chance of aging. Okay, and the trigger for reactive oxygen species are very number, but in general, if there is a high ROS, then it will lead to aging. So low ROS is incorrect. It should be high ROS. Okay, so option fourth is correct over here, but which is not a hallmark. So low ROS is incorrect. Which of the following disease is the consequence of too little apoptosis? Apoptosis we talked. Parkinson's disease, AIDS, stroke. We talked about that. If there is a high number of apoptosis, then it leads to this all the three diseases. Okay. But in autoimmune disease, there was a very little dose. Also cancer. So autoimmune disease and cancer are two examples in which too little apoptosis are present. The correct answer is autoimmune disease. Which of the following proteins can physically go to mitochondria to promote the release of the cytochrome C? We already discussed Bax and Bac. Okay. Bax and Bax are the protein. Bax and Bac are the protein which directly go to mitochondria to promote the release of cytochrome C by forming the pores in the mitochondria. Among the following proteins, which protein inhibits apoptosis? BCL2, BCL-XL, STAT1, E2F2. The correct answer will be BCL2 over here. Okay, BCL2 will inhibit the apoptosis in this case. Is this clear to everyone, or is there any doubt in other option? So, which of the following is a proto-oncogene? Proto-oncogene, there are different examples of the proto-oncogene. So, let's discuss about the proto-oncogene. Then we can go through this slide. Okay. So, proto-oncogene, as I discussed, that it leads to the gain of function mutation. Okay, that is dominant form. So, if the proto-oncogene is active, then it will lead to promotion of tumor. Okay, cancer. And another type is tumor suppressor gene, as we discussed earlier as well. That if that gene is active, then it will lead to decrease in the tumor. In the cancer, generally the proto-oncogenes are active form, and tumor suppressor genes are inactive form. So these are the examples. Uh, these are the example. RASH, SRC, MYC, ABL, BRAF. And for tumor suppressor gene, it is retinoblastoma, RB, P53, APC, BRCA1, BRCA2, and INC4. Okay. So tumor suppressor gene generally leads to the loss of function. Okay. And proto-oncogene leads to the gain of function. So these are the two examples. So now if you go to these questions, which of the following is a proto-oncogene? Retinoblastoma is a tumor suppressor gene. P53 is also a tumor suppressor gene. BRCA1 is also a tumor suppressor gene. And CMYC is a proto-oncogene. So option 4 is correct. CMYC. Okay. In C. elegans, which of the following proteins will prevent the apoptosis? Prevention of apoptosis, what we discussed. What was the pathway over there in the C. elegans? This was the pathway. CED4 was activating CED3 and it was undergoing apoptosis. And CED9 was inhibiting this CED4 and 3 complex. Okay, CED4 and CED3. So CED9 act as an anti-apoptotic molecule over there. Okay, so CED9 is the correct option in this case. Pick the correct statement about the cancer cell. Abnormal unrestrained proliferation. Yeah. Pick the correct statement about the cancer cells. Abnormal unrestrained proliferation, invasion of surrounding normal tissue, cancer cells have somatic mutation. So is this option correct? Abnormal unrestrained proliferation. This is correct option. Invasion of surrounding normal tissue. Yes, due to metastasis, it leads to the invasion. And cancer cell have somatic mutation. So this is also correct. The correct option is all of the above. Okay. Now, this option, pick the correct statement regarding the caspase-3. What is caspase-3 role? Okay, causes DNA fragmentation by activating DNase. This is correct. Caspase-3 leads to DNA fragmentation. Dephosphorylate myosin regulatory light chain. Is this correct? Does caspase-3 leads to dephosphorylate myosin regulatory light chain? Hello, is anyone there? You can directly call. Okay, you're typing them. Yeah. Yes. People is raising the. You can call. I mean, on the audio and then ask or tell what you want to tell. Yeah, anyways. So is this option correct? Dephosphorylate myosin light chain. We'll see. Third option is activated by ROC1 kinase. Is this correct? Activated by ROC1 kinase. And then activated caspase leads to membrane blebbing. So this is correct. Activated caspase leads to membrane blebbing. Option 1 and 4 is correct. Okay. And if you go through the options, then 1 and 4 is only available in second. The second option is correct. And it tells the third option is also correct. Activated by ROC1 kinase. So this mechanism we discussed, and I will again show you how this mechanism is there if you remember how caspase 3 is activating the apoptosis by this mechanism. Okay, caspase 3 will lead to the degradation of ICAD and release the caspase activated DNase, and it will lead to DNA fragmentation. Further, caspase 3 will lead to the ROC1 cleavage. Okay, it will consider the active ROC1. ROC1 then further phosphorylate MLC. Phosphorylated MLC will lead to membrane blebbing. So it is MLC phosphorylation leads to membrane blebbing, not the dephosphorylation. Okay. So what is three question? Question number three. You can directly talk, okay, because I have to like uh, go through the chat by opening. Directly you can on the audio. Question number three. Yeah, question number three I have not included over here. It was regarding the that nutrient sensing pathway uh, in the aging process. Because I have not discussed in that in this, so I have not included over here that question. Okay. So in the next class, when we solve the current assignment questions, okay, regarding to this week assignment, there we can discuss about that nutrient sensing pathway because currently the time actually is up. So that thing we can discuss that question third, okay, nutrient sensing pathway. We will first go through the theory and then we will solve that question as well as the assignment question from this week as well. Okay, which constitutes. So in the next week, I will remember and we will discuss in the next week class. Yeah. Any doubt? Others?
Sir, I have a doubt.
Yeah.
So when I
Mm.
see the progress, so since I have watched all the videos and sometimes I have watched for a couple of times while while answering the questions in the assessment, but my unit progress, the lesson progress is showing in some of the weeks it is 80%, 75%, like that. So only 2 weeks I have seen 100%. Is there any chances that it will reflect on my result?
No. If you have submitted the assignment questions, that only will count the assignment marks on the result. So if you have gone through the result part, that how it will be evaluated. So certain portion is the online exam, the offline exam which will be applying, and other like 20-30%, I don't know the exact percentage. That is 20 to 30% is from assignment. So if you have gone through the video or not gone through the video, it does not matter. It is just an indicator for you. Okay, that
Okay,
how much is the
okay.
this
And one more
score.
thing, see, I have opted for cell biology and crop breeding. So cell
Okay.
biology, almost I am attending all the interactive sessions, but crop breeding, I have not seen any interactive sessions which is conducted. I think only one interactive session was being conducted. Is there any idea that you have any communication between crop breeding faculties?
No, actually, I am only connected with this cell biology only. So other courses, I will not be able to tell whether the sessions are ongoing or not. For that, if you have certain queries for that, then you can directly mail to that faculty or the email ID of the NPTEL. You can
Okay.
type your concern over there and you can. So there is no tutorial sessions going on for that course.
No, no, they are sending the video lectures as for cell biology the same way, but there is no
Okay.
weekly interactive sessions as far now.
Okay. So maybe they might have not included the weekly interactive sessions. I don't know, but
Yes, yes.
you can like directly communicate to the NPTEL team. They will be able to answer that.
Okay. And
Yeah.
one more thing. So since this is an 8 week course, so I think it
Mm.
is the six assessments best is taken considered, isn't it, sir?
Yes. Yes. Yes.
Okay. Okay. One more
And
thing.
finals. Yeah.
So from the eight assessment questions, so, how much of percent of questions are going to repeat in the exam, sir?
That totally depends on who sets the question paper. Okay. We are not involved in setting the question paper, so I will not be able to answer that. But most of the question will be repeated. Most of the assignment question, if you have gone
Mm,
through the assignment questions, or
mm,
most of them will be repeated. But I cannot guarantee that what percentage of the questions will be repeated.
okay, not a problem. Thank you, sir.
Yeah. If you have gone through this tutorial sessions and if you have gone through, if you have basic knowledge of the course, then you might easily apply to that, easily answer that questions.
Yes, yes, sir, exactly.
And most of them would be in the multiple choice question based on almost all. So
Mm-hmm. Mm-hmm. Yes, sir, thank you.
yeah, so this was the some model questions regarding the for this today's lecture. From the previous lecture, previous histones and nucleosome formation lecture, the assignment which you have uploaded, the assignment questions which you have submitted, uh, we can discuss it in 5 minutes. Okay, the probable answers and the why that option is correct. After that, we can end the session. And if you have any doubt in any of the question, you can directly ask. The first question was in the beat order string structure, the beats are nucleosome core particle. Okay, nucleosome core particles are called beads, which are wrapped onto the DNA. That is called string. Okay. Second question is the nucleosome core particle contains a histone octamer. Okay, the histones are arranged in octameric form. Total four histones which are arranged in a two-fold axis. Okay. SIR2 is a H tag. Okay, histone deacetylase. We discussed about SIR2 that it will lead to deacetylation of the histone modification, and then it leads to the activation of the transcription of that gene. So it is an H tag. Pick the incorrect statement about SMC protein. SMC protein, as we discussed, that SMC family of protein contains two main proteins, cohesin and condensins. And apart from that, SMC45 was there, okay, which helps in the DNA repair process. So SMC protein we discussed, they are conserved. They can fold back onto themselves into antiparallel interaction, okay. They can interact directly with each other into antiparallel ways, interaction. Two SMC protein interact with each other at the hinge region, okay. At the hinge region, we talked. So SMC protein, if I tell, then this was the structure of SMC, and over here non-SMC protein were present. Okay, so at the hinge region, they were interacting with each other. Okay, so this option is also correct that two SMC protein will interact at the hinge region. So here they have asked about incorrect statement. Okay, so all these three options are correct. So none of the above will be the correct option. Okay, so you need to read the questions very carefully that what is asked. Okay, incorrect is asked and all three are correct. So none of the above will be the correct option. Tags are topologically associated domains, and we also talked about topological associated domains in the previous class. That what are topological associated domains? Basically, it helps to bring the enhancers and promoters close to each other if they are far apart from each other in a single chromosome. Whether it is topologically associated domains. What is a histone fold? Histone fold, we discussed that uh, two histones will link to each other by each other through a handshake interaction. Okay, and one histone fold consists of three alpha helices connected by two loops. Okay, so this was the kind of structure. So here there was one alpha helix, then there was a loop, then again there was one alpha helix, and again one loop, and there again there was one alpha helix. There was three alpha helix and two loops present. This is called histone fold. Okay, so third option is correct over here. Three alpha helix connected by two loops. Which of the following modifications are found in histone? This we already discussed in the initial at the start of the lecture. Acetylation is there, methylation is there, phosphorylation is there, citrullination is there. So all of the above answer is correct. To count the number of SMC or condensing complex at the loop neck during the double theta DNA assay, which fluorophore was used? So double theta DNA assay we discussed, okay, in the earlier class, and over there we use the Atto 647N dye, if you remember, to tag the condensing molecule. Okay, so to see whether at which side of the DNA condensing is binding. Okay, so for that we tag the condensing by the Atto 647 dye at their N terminal end. Okay, to visualize the DNA molecule by the fluorescence microscope. So this is a fluorophore. So option A is correct. Which of the following statement is not true? Sister chromatid cohesion promotes proper bipolar attachment by steric constraint and tension. This is correct. We already discussed that cohesion between the two sister chromatid will promote the proper tension and steric constraint so that unequal segregation of chromosomes will not happen. Okay, and bipolar attachment, proper bipolar attachment will occur by the chromosome. Okay, during the later stage of the mitosis. Without condensation, lagging chromosome can be cleaved during cell division. This is also correct. Okay, okay, so if the chromosome will not condense enough, then the part of the chromosome which is not condensed, there is a chance that during the cell division that region can be cleaved. Okay, so proper condensation is important in that case. Cohesin complex generates sister chromatid cohesion by tethering sister chromatids. This is also correct that The cohesion generally helps in the sister chromatid cohesion. Okay, two sister chromatids are cohes to each other by cohesion complex. Here it is as which is not true. All these three options are true, so none of the above options is correct. Okay. So in this case, all the three options you need to have an idea about all the three options. Okay. Then only you will able to answer this question, this type of questions. Okay. If you don't have idea about any one of the option, then you will be having doubt which of the option is correct. So all no, all the option knowledge should be there in this case. Cohesion complex is sumoylated by these are the three MCD1, SSC3, NSC1, NSC2, MMS21. So this option is correct, NSC2, MMS21. So this actually there is a regulation, okay, with the later stage cohesion complex. We also talked regarding this, okay, and during the SMC discussion, SMC complex discussion, that this MMS21 is generally sumoylated, okay, sumoylated by the protein, uh, and this sumoylation then further leads to the disintegration of that non-SMC subunit from the SMC45. So this MMS21 is generally present in the SMC45 complex, and when MMS21 or NSC2 is sumoylated, then it will lead to the disintegration of that protein. Okay, the correct option is this one, NSC2. Okay, so that's all from this class. And if you have any doubt in any of the questions or the content which we discussed, then you can ask. Otherwise, we can end the class.
Thank you so much, sir.
Okay, okay, thank you, everyone.